My name is Madison Wright, and I'm currently a fourth-year graduate student in the Plata Lab at Vanderbilt University. My work focuses on understanding protein-protein interactions that are implicated in protein folding, processing, and secretion from the cell, a process cumulatively referred to as protein quality control. More specifically, my project focuses on the thyroid prohormone, thyroglobulin, which is implicated in the protein folding disorder congenital hypothyroidism. Using the model and workflow detailed in the bottom left panel has allowed me to identify and quantify changes in protein-protein interactions that take place from mutant to wild-type thyroglobulin. In particular, we've been able to identify altered engagement with degradation machinery within the cell that suggests altered processing of mutant thyroglobulin through alternative degradation pathways. Additionally, we noticed significant changes in engagement with N-glycosylation components, particularly glycan trimming and modifying enzymes, as well as glycoprotein-specific lectins and accessory proteins that exhibited increased engagement with mutant thyroglobulin compared to wild type. What stood out to us, though, was a subtle but significant change in mutation-specific engagement with the furthest most upstream N-glycosylation component, the oligosaccharyltransferase, or OST complex. To investigate the implications of these changes, we chose to follow knockout experiments, where in the case of one mutation, A2234D, knockouts of either isoform of the OST complex significantly decrease A2234D degradation, indicating that engagement by both isoforms is necessary for the efficient degradation of this particular mutation within the cell. The work presented here has allowed us to develop a model wherein the case of mutant thyroglobulin, increased chaperoning leads to the retention of thyroglobulin within the ER, which blocks traditional degradation of the mutant protein that may otherwise be degraded, but also prevents downstream secretion. Here we presented some of the first work not only identifying a comprehensive map of the thyroglobulin interactome, but detailing protein processing mechanisms and functional implications of these interactions as it relates to congenital hypothyroidism. Moving forward, my work will focus on developing a new quantitative mass spectrometry-based method to identify these protein-protein interactions on a time-resolved scale. While there's a number of methods to identify these interactions, all of them lack temporal resolution. And one can imagine that identifying these interactions in a time-dependent manner would reveal previously unseen or poorly understood mechanisms of protein processing and could additionally aid in other biological investigations. With that, my acknowledgments are in the bottom right, and if you have any questions or comments or just want to connect, I'd be happy to chat with you. Our lab website is plata-research.org. My contact info can be found there. Thanks.